Today, we are doing something a little different. Usually, when we look at biology on this show, we look at the things that keep us alive, right? The heart, the brain, the immune system. We look at the systems working for you.
Right, the constructive biology.
Exactly. But today, we're looking at the things designed to take you apart.
And not just take you apart, but do it with a level of mechanical precision that is honestly sort of terrifying when you really look at the schematics.
We're talking about the hidden war in the human gut. And I want to be clear about the stakes right off the top here. I think when most people hear stomach bug, they think of a nuisance. You know, you eat some questionable potato salad, you take a day off work, you watch Netflix, and you drink some ginger ale. It's a bad Tuesday.
Yeah, it's seen as a minor inconvenience for most of us.
But we are talking about a specific class of viruses today that are responsible for a staggering amount of human suffering.
The numbers are, well, they're difficult to wrap your head around. If you look at the global data from our source material, specifically the chapters on Reoviruses and Caliciviruses, we are seeing that just one of these viruses, rotavirus, is linked to over 1.3 million deaths annually in preschool children in developing nations.
So that is over 1 million children every single year.
Exactly. And even in developed nations like the United States, acute gastroenteritis is the second leading cause of disease in families, right behind the common cold. But structurally, these things are light years ahead of a common cold virus. These are what I like to call the heavy industry of the viral world. They're tanks. They are armored, geometric machines that have evolved specifically to survive the harshest environment in the human body, which is acid bath of the stomach.
So here is our mission for today. We have a stack of papers covering the Reoviruses, the Caliciviruses, and a brief look at the enteroviruses. We're going to deconstruct them. We're going to look at their architecture, literally how they are built atom by atom. We'll look at the history of how we found them, which is a detective story in itself. And we're going to talk about the perfect storm of biology that makes them so incredibly contagious.
We're basically going to cover the tank, which is rotavirus, the swarm, which is norovirus, and the invaders, which are the enteroviruses.
Let's start with the tank, the Reovirus family. Now, I was looking at the etymology here because usually names in virology tell you something about the disease. You know, pox virus gives you pock marks, hepatitis inflames the liver. But Reo, that doesn't sound like a symptom.
It's not. It's an acronym. And it's actually a bit of a historical joke on the scientists who found it. Well, back in the mid-20th century, virology was really taking off. We were getting better at isolating viruses from tissue samples. We found this new family of viruses in the respiratory tract. Then we found them in the enteric tract, the gut. But here is the kicker. The people we took them from weren't sick, or at least the scientists couldn't link the virus to a specific set of symptoms at that time.
So you have a virus with no disease.
Right. It was a virus without a home, a virus without apparent disease. So they called them orphan viruses, respiratory enteric orphan viruses, REO, reovirus.
That's surprisingly poetic, the orphans of the gut.
It is. But as it turns out, the orphan label was a bit premature. While some members of the family are relatively benign, the family includes the rotavirus, which we now know is the single most important cause of severe gastroenteritis in children worldwide. It wasn't an orphan, it was a serial killer we just hadn't fingerprinted yet.
Let's talk about the mugshot of this killer then. If I put a reovirus under an electron microscope, what am I looking at? You called it a tank earlier?
I completely stand by that. To understand why, you have to compare it to something else. Think about the flu virus or COVID-19. Those are enveloped viruses. They are wrapped in a layer of fat, a lipid membrane that they stole from the host cell when they burst out.
Okay.
That fat layer is their weakness. Soap dissolves fat. That's why washing your hands works so well on them. But more importantly for this deep dive, stomach acid tears that fat apart. If you swallowed a flu virus, your stomach acid would dissolve its coat and neutralize it instantly.
So an enveloped virus is like a soldier wearing a heavy coat. If you take the coat away, they freeze.
Perfect analogy. But reoviruses, they are non-enveloped. They don't rely on the stolen coat. Instead, they build their own armor. They have a capsid, a protein shell. But not just one. Reoviruses often have a double or even triple concentric shell.
Like a Russian nesting doll.
A Russian nesting doll made of Kevlar. This structure makes them incredibly chemically stable. You can dip them in mild acids, you can dry them out on a surface, you can expose them to detergents, they survive. They are built specifically to transit through the stomach, which is basically a vat of hydrochloric acid designed to destroy proteins and arrive in the small intestine completely intact.
That is terrifying engineering. But the weirdness doesn't stop at the shell, right? It goes down to the genetic code itself. The source material keeps mentioning dsRNA.
Yes, and this is the part that makes virologists sit up straight. Most RNA viruses, remember RNA is the instruction manual for the virus, carry their code on a single strand. It's like a single long scroll of paper. It's efficient, but it's flimsy. Real viruses have double-stranded RNA.
2 strands, like DNA.
Similar structure to DNA, yeah, but with RNA chemistry. But here's why that really matters. Human cells never have double-stranded RNA floating around in the cytoplasm. It essentially doesn't exist in our normal biology.
So if a human cell sees double-stranded RNA,
it's an immediate air raid siren. We have ancient evolutionary alarm systems inside our cells. We have sensors like TLR3, which is toll-like receptor 3, or RIG-DI. These are proteins that do nothing but patrol the cell looking for double-stranded RNA.
and if they find it,
They don't ask questions, they assume it's a viral invasion, and they trigger the interferon pathway. It's the scorched earth protocol. The cell shuts down protein production, alerts the neighbors, and often initiates apoptosis cell suicide. It kills itself to stop the spread.
so the reovirus is carrying around a giant neon sign that says, I am an invader.
Exactly, which creates a massive strategic problem for the virus. How do you replicate your double-stranded genome without setting off the alarm?
And the answer leads us back to that Russian doll structure.
It does. The virus is paranoid. It never uncoats completely. Even when it's inside your cell replicating, it keeps the double-stranded genome hidden inside the inner core particle. It turns that inner shell into a safe room.
Wait, so the factory is inside the shell.
Yes. It brings its own photocopy machines, its own polymerase enzymes, specifically VP1 and VP3 inside the core. It pulls in raw materials, the nucleotides, through tiny pores in the shell, makes copies of the RNA, and then extrudes the new single-stranded messages out through the pores like a ticker tape. The cell never sees the double-stranded master copy. It's completely hidden in the safe.
That's incredibly sophisticated stealth. It's operating a black site factory inside our own cytoplasm.
It's a brilliant evolutionary workaround. If it ever let that dsRNA out, the game would be over.
Okay, let's zoom in on the most famous member of this family, the rotavirus. We know it's a reovirus, so it has the double shell and the double-stranded RNA. But visually, it's iconic.
It's beautiful in a deadly sort of way. The name comes from the Latin rota, meaning wheel. When they first looked at it under the electron microscope in the 70s, it looked like a distinct wheel with a wide hub and short spokes radiating out to a rim.
A wheel of death.
A very specific wheel of death. If you look at the genome, and this is key for understanding how we fight it, it's not just one continuous strand of dsRNA. It is segmented.
Segmented.
Think of it like an encyclopedia set. Influenza is like this too. Rotavirus has 11 distinct volumes or segments of RNA. Each segment codes for a specific protein. And scientists can actually run these on a gel-doing gel electrophoresis to see a specific barcode pattern of these 11 segments, which lets them easily distinguish a monkey strain from a human strain.
Why does it matter if it's one scroll or 11 books?
Because of sex, or, well, the viral equivalent of sex. If you have 2 different strains of rotavirus, say a human strain and a cow strain, and they both infect the same cell at the exact same time, they unpack their books in the same room. When they go to pack up the new viruses, they can accidentally swap volumes.
So the new virus comes out with volumes 1 through 10 from the human virus, but volume 4 from the cow virus.
Exactly. This is called reassortment, and it is a total nightmare for immunity. Your body might recognize the human parts, but that new cow volume 4, you've never seen it before. Your antibodies don't stick to it. This allows the virus to radically shift its appearance overnight. It's how epidemics start.
This explains why we can't just wipe it out once and be done with it. It's constantly shuffling the deck.
It is. And to understand how it attacks us, we need to meet the crew, the proteins. The source material lists a few acronyms, VP and NSP.
VP is viral protein. NSP is non-structural protein.
Correct. The VPs are the bricks of the tank. The NSPs are the tools the tank uses once it lands. There are 2 VPs you really need to know, VP7 and VP4.
VP7 and VP4. Why them?
Because they are the outer armor. VP7 is the main outer shell, the rim of the wheel. VP4 is the spike that sticks out of the shell. Imagine the tank has grappling hooks. That's VP4.
Okay, grappling hook.
These 2 proteins are the targets for our immune system. If we can make antibodies that gum up the grappling hooks, the VP4, or cover the shell, the VP7, the virus is neutralized. That's the whole goal of our immune response and of a vaccine. But VP4 has a really nasty trick up its sleeve.
I'm sensing a theme here. Nothing is simple with this virus.
No. When rotavirus is released from a host, the VP4 spike is actually in a precursor state. It's floppy, it's not fully active, it's like a grenade with the pin still in.
So it can't explode.
Not effectively. To become fully infectious, it needs the pin pulled. And do you know who pulls the pin?
Don't tell me we do.
We do, specifically our digestive enzymes. The gut is full of an enzyme called trypsin, which we use to digest protein in our food. Rotavirus has evolved to use trypsin to cleave or cut the VP4 protein. This cut snaps the protein into its rigid attack mode shape.
That is surprisingly arrogant of the virus. It relies on the very thing we use to digest food to weaponize itself.
It guarantees that the virus only activates in the gut. It doesn't want to activate in your saliva or on your skin. It waits until it is deep in the intestine, surrounded by trypsin, and then snap it, arms the warheads.
Wow. So the tank survives the acid, gets into the small intestine, and uses our own digestion to arm its grappling hooks. What happens when it actually hits a cell?
This is the replication cycle, and it has one more twist involving that orphan history. Usually, viruses enter a cell by getting swallowed in a bubble called an endosome. The cell then tries to fuse that bubble with a lysosome.
The lysosome is the trash compactor, right? It's full of acid and enzymes to destroy garbage.
Right. Most viruses panic at this stage. They try to dissolve the bubble and escape into the cytoplasm before the lysosome arrives. Rotavirus. It waits for the lysosome.
It wants to go to the trash compactor.
It needs the calcium drop and the enzymes in the lysosome to strip off that outer layer we talked about. Remember, the transcription machine is inside the inner core. The virus effectively uses the host's defense mechanism to unbox itself. It sheds the outer tank armor, leaving the active inner core to drift into the cytoplasm and start the factory.
I'm starting to see why you call this a hidden war. It's basically viral judo. It uses our momentum against us at every single step.
That's a great way to put it. Once that core is active, it starts pumping out mRNA. And this leads to the formation of something called a viroplasm.
Viroplasm. Sounds like something out of Ghostbusters.
It acts like a little viral city. It's an inclusion body, a distinct zone in the cell fluid where the virus concentrates its proteins and RNA. It walls off a section of the cell and says, "This is mine now." Inside that zone, it packs the RNA into new cores.
And this is where it stays hidden.
Yes. It packages the genome into the core before it finishes the outer shell. It ensures double-stranded RNA is never exposed naked to the cell. And then, in a very weird final step, these new cores bud into the endoplasmic reticulum, the ER, and they actually gain a temporary envelope.
Wait, I thought they were non-enveloped.
They are. It's a transient envelope. They put on a fat coat just to pass through the ER, and then they immediately take it off before leaving the cell. It's a highly unique mechanism just to finalize their assembly.
Okay, so we have this machine replicating in our gut cells, millions of copies. But as a listener, you're probably thinking, okay, cool biology, but why does this make me vomit for 3 days? How does this molecular activity translate to the actual disease?
This is where we move from structure to pathogenesis, the mechanism of disease. And there are actually 3 different ways rotavirus attacks you simultaneously. It's a combined arms approach.
Give us the breakdown.
Attack vector 1, malabsorption. Rotavirus specifically infects the enterocytes at the very tips of the villi in your small intestine.
The villi are those little shag carpet fingers that absorb our nutrients.
Exactly. Rotavirus kills those cells. It effectively mows the lawn of your gut. You lose the mature cells that are supposed to absorb water and sugars, and you also lose the digestive enzymes they produce, like maltase and sucrase. So water stays in the gut, and undigested sugar stays in the gut.
And osmosis dictates that water follows sugar.
Right. So water's pulled out of your body and into your gut to dilute that sugar. That causes osmotic diarrhea. But that's standard. That's what happens if you just eat something you can't digest. Rotavirus does something else. Attack vector 2, the enterotoxin.
This is the NSP4 protein mentioned in the text.
Yes, and this requires a deep dive because it was a massive paradigm shift in virology. For decades, we thought viruses cause diarrhea just by killing cells. Bacteria have toxins like cholera toxin that actively pump water out. But viruses, we didn't think so until we found NSP4.
What does NSP4 actually do? How does it work?
NSP4 is a viral protein, but it acts like a signaling molecule. It acts like a bacterial toxin. It binds to receptors on the gut cells and triggers a signal transduction pathway. Specifically, it causes a massive release of calcium from the endoplasmic reticulum inside the cell.
Okay, so calcium floods the cell.
Calcium is a universal on switch for cells. This calcium surge disrupts the cytoskeleton, the scaffolding of the cell, breaking the tight junctions between cells, so the gut becomes leaky, but it also stimulates the enteric nervous system.
The gut has a nervous system.
A very complex one. NSP4 essentially hacks it. It triggers the secretion of chloride, and where chloride goes, water follows. So even the cells that aren't infected are being chemically ordered to dump water into the intestine.
So it's not just that you can't absorb water because the lawn is mowed. The virus is actively pumping water out of your body via a chemical signal.
Precisely. It turns the gut into a flowing river, which, if you are a virus, is exactly what you want.
Because you need to get out to find a new host.
The diarrhea is the transport mechanism. It flushes billions of viral particles out into the world to find the next victim. It's incredibly efficient fecal-oral transmission. And that leads to attack vector 3, stimulation of the vagus nerve.
The vagus nerve connects the gut to the brain.
And specifically to the vomiting center of the brainstem. The release of serotonin from these damaged gut cells travels up the vagus nerve and triggers the violent vomiting reflex. This is often the first symptom.
So you have liquid exiting both ends.
And rapid dehydration. In a 180-pound adult, it's miserable. In a 15-pound infant, it's catastrophic. You can lose a dangerous percentage of your body water in hours. That leads to electrolyte imbalance, cardiac arrest, and death. That's how you get to that 1.3 million deaths figure globally.
It's sobering to realize that all of this sophisticated machinery, the double shell, the trypsin activation, the calcium signaling, is all designed just to create that flow of water.
It's an extraction machine. It extracts water and it extracts itself.
Which brings us to the obvious question. If this thing is a tank that hacks our nervous system, how do we stop it? We mentioned reassortment makes it hard. But we do have vaccines now.
We do, but the road to getting them was, let's call it tragic. It's one of the most dramatic stories in modern public health history.
You're talking about Rotaviruses.
Yes. In the late 90s, science had finally cracked it. We had a vaccine. It was a live attenuated vaccine, meaning it used a weakened version of the virus. It was licensed in the US in 1998 with huge fanfare. Pediatricians were relieved. Finally, we could stop this winter scourge.
And then the reports started coming in.
It took about a year. Doctors started noticing a strange correlation. Infants who had just received the vaccine were showing up in ERs with intussusception.
For those who don't know, what is that?
It's a mechanical bowel obstruction. Basically, one part of the intestine slides inside the adjacent part, like a telescope collapsing on itself. It cuts off the blood supply. The bowel can die. It is a surgical emergency.
God, and it was caused by the vaccine.
The statistical link was undeniable. It was rare, about 1 in 10,000 cases. But for a vaccine given to healthy babies, that risk is entirely unacceptable. The vaccine was pulled from the market in 1999.
That must have been a devastating blow to the research community.
It was heartbreaking. It set the field back years. But to their credit, the scientists didn't stop. They went back to the drawing board. They realized they needed a different approach. And this is where that reassortment trick we talked about, the gene swapping, actually became the solution.
How do you use the virus's weapon against it?
They looked at animal rotaviruses. Cows get rotavirus, but cow rotavirus doesn't make humans sick. However, it looks enough like the human virus to wake up the immune system.
So the Meteor approach.
Yeah.
Like cowpox and smallpox.
Exactly, but with a twist. They created Rotatech. They took a cow rotavirus strain, but they used genetic engineering, specifically reassortment in the lab, to swap in the outer spike proteins, the VP4 and VP7, from human viruses.
So it's a wolf in sheep's clothing. Or a cow in human's clothing.
Yeah. A cow tank painted with human colors. To the human immune system, it looks like the deadly human virus, so we make antibodies against those VP4 spikes and the VP7 rim. But inside, it's a cow virus. It replicates poorly in humans so it doesn't cause the disease.
That is brilliant.
It's the basis of the pentavalent vaccine we use today. There's also Rotarix, an attenuated human strain. And since their introduction around 2006, intussusception rates haven't spiked, and deaths from diarrhea have plummeted globally. It is a massive victory.
It really highlights the persistence of science. One failure didn't end the war. Now, the source material briefly mentions another branch of the reovirus family, the arboviruses. like orbovirus and cultivirus.
Right. It's a good distinction to make. Not all reoviruses cause diarrhea. Cultivirus, for example, causes Colorado tick fever.
Transmitted by ticks, obviously.
Yes, the wood tick. Mostly in the southwestern U.S. and mountainous regions. It causes fever, rash, and severe muscle pain. It's very ding-like. And it's sneaky. It infects red blood cell precursors, allowing it to hide inside the red blood cells and stay in your blood for weeks.
But they are the exception to the gut rule for today.
Right.
So rotavirus is just one fighter in this enteric arena. We have to pivot now. Because if rotavirus is the tank designed for the long siege of the intestine, the next virus on our list is the swarm.
You're talking about the caliciviruses.
And specifically the norovirus.
Yes.
Okay, norovirus. The winter vomiting disease, the cruise ship curse. Before we get into why it's so contagious, let's look at the cali part of the name.
It comes from the Latin calyx, meaning cup. If you look at these viruses under an electron microscope, they have these characteristic cup-shaped depressions on their surface.
And how do they compare to the rotavirus tank?
They're smaller, about 27 to 40 nanometers. They are single-stranded RNA, positive sense. And like rotavirus, they are non-enveloped. But unlike rotavirus, which relies on that complex double shell, norovirus relies on sheer numbers and stability.
We call it the perfect pathogen. Wow.
It's the perfect storm of three factors. First, the infectious dose. This is the number of viral particles you need to ingest to get sick. For some bacteria, like Salmonella, you might need to eat 100,000 bacteria to get sick.
And for norovirus?
As few as 10.
10 particles.
10. You could fit 10 million particles on the head of a pin. 10 is essentially invisible. If someone vomits in a room, the aerosol spray can carry thousands of particles to the other side of the room. You breathe in 10, you swallow them, you are infected, and the incubation is short, 24 to 48 hours.
That is terrifyingly efficient. What's the second factor?
High shedding. When a person is sick with norovirus, they are shedding billions of viral particles in their stool and vomit. So you have a virus that requires almost nothing to infect you, and the host produces an infinite supply of it.
And a third?
Durability. Norovirus is the cockroach of the viral world. It is incredibly tough. It resists chlorine bleach at concentrations that would kill almost anything else, up to 10 parts per million. It survives freezing. It survives heating to 60 degrees Celsius.
So you can't freeze it out, and you can't wash it away easily.
Exactly. It persists on surfaces, door handles, railings, elevator buttons for weeks. And it bioaccumulates in filter feeders. Oysters are the classic vector. They sit in contaminated water, filtering gallons a day, and they concentrate the virus in their tissue. If you eat a raw oyster that has norovirus, you are basically taking a shot of pure virus. Even steamed oysters sometimes aren't cooked enough to kill it because the virus is so heat stable.
Remind me to order the fried clams instead.
Yeah.
Well, I want to get to a specific piece of listener feedback we received. We talked about how contagious this is. But I have a friend who swears he never gets the stomach flu. Even when his whole family's down with norovirus, he's totally fine. Is he just lucky? Or is there biology at play?
He might not be lucky. He might be a mutant. In a good way.
Explain.
This is one of the most fascinating aspects of norovirus biology. It involves your blood type and something called secretor status.
Secretor status.
So we all know about A, B, and O blood types. Those are determined by sugars on the surface of your red blood cells. But most people, at 80% of the population, also secrete those same sugars on the surface of the cells lining their gut. These people are called secretors.
And the other 20%.
They are non-secretors. They have a mutation in a gene called FUT2, fucosyltransferase 2. This gene is responsible for building the little sugar docking station on the gut cell. If you have the mutation, you don't build the dock.
And norovirus needs the dock.
Exactly. Norovirus uses those specific blood group antigens as receptors to bind to the cell. If you are a non-secretor, your gut cells are like a smooth wall with no handles. The virus literally cannot grab on.
So if you are a non-secretor, you are naturally immune to norovirus.
To the most common strains, yes. It's a genetic shield. So if your friend never gets sick, he probably has that FUT2 mutation.
That is a legitimate superpower. Can I get tested for that?
You can, actually. Commercial genetic tests often report on it. But be warned, norovirus evolves. There are some strains that might find other ways in, but generally, non-secretors are the ones walking around healthy on the plague ship.
Speaking of evolution, why don't we have a vaccine for this? We have one for rotavirus.
It's tricky. First, we can't grow norovirus in the lab very well. Rotavirus grows in cell culture, and norovirus is notoriously fussy. If you can't grow it, you can't easily study it or make a vaccine from it. Second, the immunity is weird.
Weird how?
With rotavirus, once you've had it a few times, you are pretty much protected for life against severe disease. With norovirus, immunity lasts maybe 6 months to 2 years, and then it fades. Plus, the virus drifts, it changes its coat.
So it's like the common cold. You can catch it over and over again.
Exactly. There are different genogroups, GI, GII, GIV, and within those, countless strains. We are working on vaccines. Some use virus-like particles, which are empty shells that look like the virus, but we aren't there yet.
So for now, the best defense is
Bleach and vigorous hand washing. Alcohol like hand sanitizer actually doesn't work very well on norovirus.
Wait, really?
Really? Remember, alcohol works by dissolving the fat envelope of viruses like flu or COVID. Norovirus is a non-enveloped cup. The alcohol just slides right off. It needs to be physically washed away with soap and water.
That is a crucial public service announcement. Hand sanitizer is not a magic shield against the stomach flu.
It is not.
Okay, we've done the tank, which is rotavirus, and the swarm, norovirus. I want to briefly mention the astrovirus.
The star, astra means star in Greek.
These are the quieter cousins.
They are. Under the scope, they have a 5 or 6-pointed star shape on their surface. They're tiny, 28 to 30 nanometers. They cause mild gastroenteritis, mostly in infants and the elderly. We don't talk about them as much because they don't cause the massive death toll of rotavirus or the explosive outbreaks of norovirus, and they're really hard to grow in the lab, so they often get missed. But they are there, contributing to the burden of disease.
Now, before we close out, we have to address a confusing point of terminology in the source material. We've been talking about enteric viruses, viruses in the gut. But there is a specific genus called enterovirus. And ironically, they usually don't cause diarrhea.
This is a classic medical student stumbling block. The genus Enterovirus, which includes polio, Coxsackie, and echovirus, belongs to the Picornaviridae family. Pico means small, RNA means, well, RNA.
So these are small RNA viruses?
Yes. They are enteroviruses because they enter through the enteric tract. They are transmitted fecal-orally. They replicate in the gut, and this is the key, the gut is just the gateway. It's the beachhead.
They don't stay there.
No. They burrow through the gut wall and enter the bloodstream. From there, they travel to their actual target organs.
Which are?
The skin, the muscles, the heart, and the nervous system.
This is the Trojan horse strategy.
Exactly. You ingest it, it sets up shop in your intestines quietly, and then suddenly you have meningitis or heart failure. Both Group A and Group B Coxsackie viruses can cause viral or aseptic meningitis.
Let's break down the Coxsackie viruses specifically. There are Group A and Group B.
Group A tends to target the skin and mucous membranes. If you have kids, you know hand, foot, and mouth disease.
Oh yes, the blisters.
That's usually Coxsackie A16. It causes painful vesicular blisters on the palms, soles, and inside the mouth. It's miserable, but usually self-limiting. Then there is herpingina, which causes severe fever and vesicles at the back of the throat on the soft palate.
And Group B?
Group B is scarier. It targets the deeper tissues. It targets the heart and the pleura, the lining of the lungs. Have you heard of the devil's grip?
That sounds like a horror movie title.
It's the medical nickname for pleurodynia. It's caused by Group B Coxsackie. It causes an infection of the intercostal muscles, the muscles between your ribs.
And what does that feel like?
It causes spasmodic, stabbing pain in the chest. It comes on suddenly. People often think they're having a massive heart attack. It is excruciating, hence the devil's grip.
But you mentioned it can actually attack the heart itself.
Yes. Viral myocarditis. The virus infects the heart muscle cells directly. It causes inflammation. This can lead to heart failure. It can affect infants, causing generalized disease of infants, which is essentially overwhelming viral sepsis with heart and liver failure, but it can also strike totally healthy adults.
So a virus you ate ends up breaking your heart.
Literally. It's a reminder that the gut is the central plaza of the body. If a virus can master the gut, it can go anywhere.
We've covered a massive amount of biology today, from the enzyme-activated spikes of rotavirus to the genetic immunity of norovirus to the Trojan horse of Coxsackie. If a doctor suspects one of these, how do they actually tell them apart in the lab? I assume they aren't looking for wheels and stars in every patient.
Historically, they did. Electron microscopy was the gold standard. You'd take a stool sample, spin it down, and look for the shapes, but that requires an incredibly expensive microscope and a specialist.
So what do we do now?
For rotavirus, we use ELISA. It's an antigen test. Think of it like a rapid test strip, but for stool. It looks for the viral proteins. It's fast and cheap.
And for norovirus?
Because the infectious dose is so low and the shedding is so high, PCR, or polymerase chain reaction, is the better tool. We amplify the genetic material. It's incredibly sensitive. It can find those 10 particles we talked about. Serology checking for antibodies is mostly just used for epidemiology, not for treating acute sickness.
So, as we wrap up this deep dive, what is the big picture here? We have these ancient machines.
I think the takeaway is respect. We tend to view viruses as simple, but look at the rotavirus. It has a segmented genome that allows it to shuffle its identity. It has a triple-layer tank armor to survive acid. It uses a specific host enzyme, trypsin, as a GPS to know when to activate. It uses a toxin, NSP4, to hack our nervous system and flood our gut.
And norovirus.
It's the ultimate survivor. It stripped away everything it didn't need. It's just a tough, cup-shaped shell and a code that mutates fast enough to keep us guessing. It exploits our social nature, our food sharing, our close quarters.
And the enteroviruses remind us that the gut is just the front door to the rest of the body.
Exactly. These viruses have evolved alongside us for millions of years. They know our biology better than we do. They know which enzymes we have, which receptors we display, and exactly how our immune system reacts.
So the next time you hear about a stomach flu, or you're unfortunately clutching your stomach, remember, it's not just bad luck. It is a molecular battle against a specifically designed, geometry-perfect viral machine taking place inside you.
And understanding that design down to the atomic level is the only way we eventually win the war. But there is one final thing to mull over.
What's that?
We talked about how norovirus is resistant to chlorine, and rotavirus survives acid. We rely so heavily on our modern hygiene water treatment, hand washing, sanitation, but these biological machines evolved specifically to bypass the ultimate chemical barrier, our own stomach acid. It makes you wonder, as we engineer better cleaning chemicals, are we just forcing them to build stronger armor?
That is a chilling thought to leave you with. Keep exploring the invisible world, everyone.
Stay curious.
We'll see you in the next deep dive.